Hi, I'm Sam Osry, and I'm presenting over Q fever. So Q fever is a disease which generally presents itself as atypical pneumonia or an influenza-like illness in some milder cases. It's caused by the bacteria Coxiella burnetii, and this bacteria is technically gram-negative, although uh, it doesn't really work well for the gram technique. But it has a lipopolysaccharide, which is really important for its really important virulence factor for it. It's also a very, very small bacteria. In fact, when it was first discovered, it was thought that maybe it was the disease, the bacteria or the agent that was causing the disease was a bacteria because it was so small that it was going through their filter. Uh, as far as its taxonomy, it's part of the order Legionellales, um, the family Coxielaceae, the genus Coxiella, and the species Burnetii, which we'll talk about those names just a little later. Uh, Another few characteristics of this bacteria, it exists in three forms. There's a large cell variant, which is more metabolically active and is important for its replication. There's the small cell variant, which is generally more hardy, and then there's the spore-like particles. The bacteria is an acidophile. It thrives in a really low pH and replicates in a very acidic environment. It's also resistant to UV light, desiccation, heat, and is just overall very, very can survive in harsh environments over a long period of time. It's also extremely infectious. It's one of the most infectious bacteria that is known. It only requires 1 to 10 bacteria to actually cause an infection, a human infection. So it's very infectious. Some signs and symptoms of Q fever. The illness may be mild or severe, and usually symptoms develop between 2 to 3 weeks after exposure. About half are asymptomatic, and symptoms are may vary, and they're generally not very. There's not any telltale signs of Q fever, so it just presents itself as a febrile illness with all those symptoms. It may lead to atypical pneumonia, hepatitis, and also some CNS complications. There are two uh, ways in which Q fever manifests itself. The first is acute Q fever, which is what I was just talking about just now. But then there's also chronic Q fever, which occurs in less than 5% of cases, and it generally develops uh, for well after you had the initial acute Q fever. Um, Usually there are some vague symptoms such as persistent fatigue, but there can be some very, very serious complications, uh, particularly endocarditis. And then also chronic hepatitis and chronic pulmonary disease is seen in some of the chronic Q fever cases. High risk individuals would be those with underlying cardiovascular disease, the immunocompromised, and pregnant women. In regard to diagnosis and treatment, it's diagnosed through PCR or indirect immunofluorescence antibody test. And the treatment for acute illness is generally a regimen of doxycycline. Uh, at least it's what the CDC recommends. Um, and usually the acute illness will subside within 2 weeks, even on its own, without the antibiotics. But antibiotics are certainly recommended by the CDC. Uh, Treatment for chronic illness is very challenging and kind of depends on the manifestation of the disease. But oftentimes there will be a regimen of doxycycline and hydroxychloroquine, and the death rate is significantly higher for those with chronic illness. As far as transmission, It's very, very versatile. It can infect a wide range of hosts. These include domesticated animals, uh, such as cows, goats, sheep. Ruminants, in particular, are important in transmitting the disease to humans. It can also be in dogs and cats. It's also found in wild animal populations, wild mammals, wild birds, and also ticks and arthropods. So it's got a wide range of hosts. Generally, it is transmitted to humans through airborne transmission. So contaminated dust particles that are contaminated with like urine or some parturitive material, like the placenta of a ruminant, like a goat or something. Um, Less common sources would be consuming raw contaminated milk, vector transmission, so from ticks. And then there has been like one or two documented cases of sexually transmitted disease, but that's very, very rare. It's generally airborne transmission. The pathogenesis, like I said, it's transmitted through contaminated dust particles usually, and C. burnetii usually enters a host as metabolically less active small cell variant, and later converts more to the large cell variant after it successfully invades the host cell. And because the large cell variant is more metabolically active and will be important for replication. It initially targets alveolar macrophages uh, and also Kupffer cells in the liver. It may also infect epithelial and endothelial cells as it goes through the bloodstream. Uh, and a really important virulence factor for this bacteria is its lipopolysaccharide, which is used to cover surface proteins and avoid immune response from the host. It's phagocytized by macrophages and monocytes, and when it's in the phagosome, it fuses with the lysosome to become a phagolysosome. And unlike many other bacteria in which the phagolysosome would destroy it, C. burnetii thrives in this environment of the secondary lysosome. In fact, it needs this environment, this acidic environment, for its replication. And it's not totally known the complete mechanism for how it is able to survive in this environment, but it is the environment in which it replicates. It creates a parasitophorous vacuole as it replicates, and eventually the cell will lyse, and it will release bacteria and infect other cells. The discovery is kind of an interesting case. There was a cluster of cases which in Brisbane, Australia, in the 1930s, which doctors were referring to as abattoir fever because it was presenting itself in abattoir workers. And Edward Derrick of Queensland Health Department recognized it as being caused by an unknown agent, and so he sent some tissue sample to Macfarlane Burnet, who successfully isolated it in 1937. And because it was so small and intracellular, obligate intracellular pathogen, and other reasons, he originally classified it as a rickettsia. He thought it was a rickettsia. Meanwhile, Harold Cox in Montana and his team were working on trying to. They were working on certain tick-borne diseases. I think it was tularemia and Rocky Mountain spotted fever. And so they were collecting ticks in 9 Mile Camp, and these ticks actually caused a disease in the guinea pigs that they did not recognize, and so that led them to the discovery and isolation of C. burnetii, which they again originally thought was a rickettsia. So he suggested calling it Rickettsia diasporica. But in 1948, it was removed from the Rickettsia genus because it was thought to be too distantly related to Rickettsia, and a whole entire genus was created for it. It's the only species in its genus, and the genus was named Coxiella for Cox. So now it's Coxiella burnetii. Incidence and prevalence. It was made a notifiable disease in the U.S. in 1999, and it's found just about all over the globe except in New Zealand, which is kind of ironic because New Zealand is a major dairy producer, livestock producer. And the following are some graphs from the CDC. Here it shows in the cases in the U.S. Generally, it's not many cases. Less than 180. And you'll see the states where it's most prevalent are in livestock producing states. And also interestingly, usually the number of cases are highest between April and June. But there have been some notable outbreaks. One of the major outbreaks was in the Netherlands in 2007 and 2010, and it caused high rates of spontaneous abortions in goats. And there was a really dry spring, and it caused this major outbreak of 4,026 cases, documented cases. And there wasn't a single identified source, but they think it was primarily due to goat farms and the dust coming from goat farms to, coming to the city and these towns and infecting people. And so there was various government action which was taken, which was kind of devastating to the goat farmers. But yeah, it was kind of a significant outbreak. This bacteria has great potential, significant potential for bioterrorism, and it was studied by the U.S. and the USSR during the Cold War. And as far as prevention of future outbreaks, a lot of that will have to do with improved agricultural practices and and vaccination of animals and veterinary surveillance. So, and the Netherlands actually made it a requirement for vets to report any case in any animals. So that should hopefully help prevent future outbreaks if these things are done well. And that's about it. Thank you.